This presentation is by Professor Pier Giuseppe Agostoni from Milan. It is narrated by Dr. Paul Older. This is a composite graph of measurements made during a breath-by-breath cardiopulmonary exercise test on a cycloergometer. Notice, though, we also have some arterial measurements of lactate, bicarbonate, and pH. Let us then define what we're going to discuss. We use the terms anaerobic threshold, lactate threshold, and lactic acidosis threshold as if they are all different. Remember that all these terms represent a common metabolic point, and that is the point where anaerobic metabolism supplements aerobic metabolism. Anaerobic threshold is a gas exchange measurement and is the point where high-energy phosphate, derived anaerobically, supplements aerobic metabolism. This results in an increase in CO2 production. That's important, as we'll see later. Lactate threshold is the point during exercise when there's an increase in lactate in arterial blood. The lactic acidosis threshold is the point above which the arterial bicarbonate decreases, thus producing a fall in pH. Let us look at this. In 1986, Beaver, Wasserman, and Whip set a method up to detect the anaerobic threshold using a computerized regression analysis of the slopes of CO2 output versus oxygen uptake. Let's look at that. There's the CO2 output, oxygen uptake. Notice there is a slope there at the oxygen uptake, which if extrapolated would go on like that. But at this point, something happens and the slope becomes steeper and goes up like that. This slope they termed S1, that slope they termed S2, and where the two slopes intersected, they called the anaerobic threshold. This then is the anaerobic threshold by the gas measurement. Notice at that point. In the same point, the lactic acid threshold, the lactic threshold from arterial blood. Notice they are the same. Notice in this graph, further examples. Here, in this test, the lactate there, what, 700 and something, but the VCO2 and O2 slope method had it there, lactate had it there, the same. Isocapnic buffering is the period when the VE and VCO2 are both rising. There we are, there. But the VE, VCO2 is not. Let's just now look at the oxygen uptake and CO2 production. Oxygen is taken up by the lungs, transferred by the blood through to the muscle to the mitochondria, where it reacts with substrate to produce energy, ATP, and carbon dioxide, which is then excreted. If then you turn over to a point where anaerobic metabolism supplements aerobic, you still have the aerobic, but now you add to it the anaerobic. And here you form lactate, hydrogen lactate, which is lactic acid. Bicarbonate will buffer that inside the cell to form carbon dioxide, water, and potassium lactate. Lactate goes into the blood. Lactate itself is not an acid, so it forms lactate. However, oxygen producing CO2, oxygen producing CO2, but now more CO2. Let's look at this graph. This is two constant work rate tests. One of them, the open circle, represents a test below the anaerobic threshold. Remember, constant work rate tests. The solid circles are above the anaerobic threshold. Notice no difference in PO2. But in the work constant test above the anaerobic threshold, the hemoglobin saturation dramatically falls. This is produced by the change in pH. This is known as the Boer effect, which effectively shifts the hemoglobin dissociation curve to the right, allowing more oxygen to be taken up by the tissues. Let's look at some circulatory dynamics. At the start of exercise, there is an abrupt increase in both VO2 and VCO2. It's very abrupt there, same there, where there's above or below the anaerobic threshold. After a short delay, there is a further increase in VO2 and VCO2 when venous blood from the exercising muscle first arrives at the lungs. In about 3 minutes of exercise below the anaerobic threshold, the VO2 forms a plateau. It takes about 3 minutes. No such plateau occurs with a constant exercise test over the lactic acidosis or anaerobic threshold. This is a research laboratory in which Professor Agostoni at 4,500 plus meters did some exercise tests. Let's just look at them. Efficiency in ventilation decreased with the VVC slope increasing from, 28-ish up to 40 or so. Isocapnic buffing has disappeared because the respiratory compensation point in the anaerobic threshold appeared to be inseparable, so there is no isocapnic buffering. At each exercise step, the end tidal CO2 was lower. So here it's up there, what's about there, whereas here it's down about there. Is the anaerobic threshold determination of any significance in patients with cardiac failure? Well, let's look at this. This is a study done by, Professor Agostoni, looking at the prognostic value of indeterminable anaerobic threshold in heart failure. 303,000 odd patients enrolled. These were excluded as they had respiratory disorders, leaving 2,137. Of these, almost 10% did not have their anaerobic threshold determined. They couldn't find it. Well, let's look at the relevance of that. The green is the patients in whom the AT was identified, the blue where it wasn't. And you can see for each year that goes by, the survival probability gets bigger and bigger difference between the two. All-cause death, the same thing. Whatever the cause of death, you're more likely to survive if they know your AT than if you don't. If we then divide into tertiles, where the tertile is a third of the group of equal size. So each one of these are around 640, and these represent anaerobic thresholds of lower and lower value. But notice, whatever your anaerobic threshold, you're still better off if we know it than if we don't. The same thing applies to all-cause death. Well, now, you may have heard the term double anesthetic threshold, anaerobic threshold. Let's just have a look at this. First of all, is there a double anaerobic threshold in healthy patients? Well, the work done here in this article, let's look. These are 2 patients, both of them healthy, neither of them sick. The V-slope method of determination of AT is the same in both patients. There it is. However, the VVO2, which is here in this patient, is right down here in this one. Clearly, a very different point. The PeTO2, again, there, but here, a very different point. Now, that's the line of the normal V-slope method, and this is the line of the ventilatory equivalent method. As you can see, substantial difference. An overview of the study population of individuals who've shown 2 anaerobic thresholds. So there were 476 patients, and 51 showed the difference. 20 women, 31 men. Now let's look at this healthy group. Notice with no double anaerobic threshold, with a double anaerobic threshold. As you can see, there's not a lot of difference in those measurements. They're mostly insignificant. If now we look at the exercise data detected by the anaerobic threshold via V-slope compared to ventilator equivalent method, these are healthy patients with a AAT51. We can see that in all cases, the V-slope appears before the ventilator equivalent slope, and there are some significant differences in values. This then is the box and whisker plot, where we can see that if you prefer things graphically, you can see this. The V is the V-slope method, and the orange is the ventilator equivalent method. But how about the double anaerobic threshold in patients with heart failure? Here we have 174 patients. 46, 51, 57, 30. Some of the patients are on carvedilol, some are on bisoprolol. Note that these patients demonstrate a double anaerobic threshold, and so do these. But the incidence of double anaerobic threshold is much higher in patients on carvedilol than on bisoprolol. This is the exercise data in patients in heart failure after 2 months of treatment on either carvedilol or bisoprolol. You can see again that these are all statistically valid. A lot of these aren't, but these, importantly, are a marked difference. You may have heard of the MECI data score base, and this is the incidence of double anaerobic threshold in patients with heart failure, incorporating the MECI score database. The MECI score stands for Metabolic Exercise Test Data combined with cardiac and kidney indices. It's a prognostic model to assess the risk of heart failure based on a combination of various clinical parameters that you can see here. This was done by Professor Agostoni and many others. And here we can see that all these patients, 1,075, no double threshold, 610, double threshold in 465. All these patients were on some form of medical treatment. Notice these figures are highly significant. Most of the others are not. But there were 465, 1,075 who had a double threshold. Here you can see again, always the V-slope method appears before the VO2 method, is highly significant. And here you have the box and whisker plot, which demonstrates that. Remember, at any stage, you can look at this again and pause the demonstration to have a longer look at the graphs. Thank you.